I'm going to talk about alpha-synuclein post-translational modifications and subcellular localization. This is a topic of great interest because alpha-synuclein, as any protein that exists in a cell and is produced in a biological system, is a protein that undergoes chemical modifications during its lifetime. So what this means is that the sequence of amino acids that we know is encoded by the DNA, by the genome, is not enough for us to know what alpha-synuclein really is. So we know, and we've known this for a long time, that alpha-synuclein can be modified, for example, by ubiquitination, by nitration, by acetylation, in different residues. And what we don't know is what is the contribution of each of these modifications to aggregation. Of course, we have some ideas. There's been a lot of work on these modifications individually, but I think we are at the stage where we need to start understanding how having a particular modification in one residue, in one side of the protein, affects other modifications in other sites in the protein. And this is important because it could be that a combination of these modifications or a modification that has a very strong effect could change the conformation, the shape of the protein, and trigger its aggregation, which we still think is relevant in the context of pathology. So now, and for quite some time now, we've been paying attention to one of these modifications, that is the phosphorylation on serine at position 129. So alpha-synuclein has 140 amino acids, and this particular modification is on serine 129. And this was because it was found already more than 20 years ago that in Lewy bodies, in the pathological inclusions that exist in the brains of patients, this residue is phosphorylated. So there has been a lot of work, a lot of interest in understanding whether this was pathological or protective. If you look in the literature, you find all sorts of results. This depends probably on the cellular context, on the animal model that is used, and so it hasn't been so easy to really define what is the role of this modification. But some studies suggest it may actually be protective. So in analogy with this, we want to study other modifications, and in my group we've studied, for example, acetylation. Acetylation is a modification that happens in lysine residues typically, and alpha-synuclein has 15 of these lysine residues. So we found that acetylation in lysine 6 and lysine 10 seems to increase the solubility of the protein and reduce its aggregation and toxicity. On the other hand, we found that glycation, a modification that we are very interested in because we think this could explain a possible connection between diabetes and the risk of Parkinson's disease. So we think that glycation is increasing the propensity of the protein to aggregate, but to form particular types of aggregates that are not necessarily the fibrillar forms that we think are components of Lewy bodies and of glial cytoplasmic inclusions in MSA. And we saw that this modification can then promote the accumulation of oligomeric species of alpha-synuclein, which we showed in experimental models that this can be bad. So of course, now the next step is to find this and demonstrate that in the human brain, this modification is associated with pathology. So to try to achieve that, we've been developing tools that were not available, like antibodies that recognize glycated forms of alpha-synuclein, and we are now testing these antibodies in human brain tissue, and we are finding something very interesting. We're finding that they label Lewy bodies as well. So it suggests, as we were guessing, that you may detect some glycated forms of alpha-synuclein in Lewy bodies. So there's a lot of research going on in this field, in people looking at other post-translation modifications, and I think this is an area that will grow and will be very important because this may really give us ideas of how to target alpha-synuclein therapeutically, and maybe also as a biomarker, maybe we can define which modified forms of alpha-synuclein we need to target in order to use this as a possible biomarker. On the other hand, we also have to think that alpha-synuclein, as any protein, exists in the cell, and in this case we tend to think that alpha-synuclein is a presynaptic protein that associates with synaptic vesicles and compartments on synaptic in the synaptic region. But interestingly, and that's also how the protein was originally identified, the protein was found to occur also in the nucleus, but no one paid a lot of attention to this. There have been several reports by our group and other groups over the years, but this was not looked as something very important. But we think it is, because we think that depending on where the protein exists, it will of course interact with different partners, with different proteins, with different components in the cell. And this will determine whether it's doing good things or bad things to the cell, whether it's causing toxicity or whether it's performing its normal function. So it can be in the nucleus, we confirm this. We are trying to understand what it is doing in the nucleus, if it's causing, for example, changes in DNA transcription, if it's damaging DNA, and we have evidence suggesting that it may be damaging DNA, and we don't know why this is happening, so we need to study this further. And other groups have found it also in mitochondria and in membranes that connect mitochondria and ER. These are compartments in the cell that perform important functions, and therefore we think it's very important to study the precise location of alpha-synuclein in the cell, in these different compartments, because we also know that alpha-synuclein is not only a neuronal protein. So when it exists in other cell types, in astrocytes or in oligodendrocytes, even if at low levels, it will not be performing the same function that we think it performs in neurons. So we need to understand what it's doing in those cells. And another cell type where it exists is in red blood cells in the blood. And no one is studying this in very much detail. So there's a lot to learn here. And so that's why I think it's important to consider also how post-translation modifications that I talked about in the beginning, how they influence the subcellular localization of alpha-synuclein. So these two topics come together, and they're very relevant in the context of understanding the normal function, the physiology of the protein, but also the pathological roles the protein may perform in disease, in Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy.